not coming. Okay. Hello. Hello, I'd like to welcome you to the 11th Annual College of Natural and Agricultural Sciences Science Lecture Series. The title of our series this year, as you can see, is Gene Editing, Are We Playing God? It's centered around the CRISPR technology, which you might have heard about if you were here in a previous lecture. So I'm Francie Sladek. I'm the Divisional Dean of Life Sciences in our college. How many of you heard either of the first two lectures in this series? Okay, good. All right, all right. Are you all ready for a quiz? What's guide RNA? Okay, all right. How many UCR staff, alumni, or students do we have here? Okay, good number. All right, okay. What about community college students? Just a few? Okay, welcome. What about high school students? Okay, great. I'd like to give a particular call. How about John W. North School? Okay, yay, yay. Norco College? Anyone from Norco College? Murrieta Valley High School? We have Riverside Community College. Obviously, these aren't high schools anymore. Santiago High School? Oh, okay. Moreno Valley College? No, okay. San Bernardino Valley College? University Heights Middle School? No, all right. Ramona High School? Pauley High School? King High School? Okay, all right. Well, I want to welcome you all. I also want to give a thanks to our science ambassadors. They're the students in the blue t-shirts. These are some of our top students in our college, and they're here to assist you with this event. I'd also like to thank 91.9 KVCR that keeps us all informed about news and other things on a daily basis from NPR. And CAFE, the California Agricultural and Food Enterprise. They're helping to sponsor this program tonight. If you have not received a t-shirt, we have some free t-shirt giveaways. And if you didn't pick one up on the way in, you can pick one up on the way out after 7 o'clock. But please, evidently these are really popular t-shirts. They're very, very nice. It has CNAS written in the periodic table of elements. And some people like them so much they take more than one or two or three. So please, we do have limited supplies. So if you already have one, don't pick one up. But if you haven't gotten one, you can get one after 7 o'clock today. So the title of the talk today is Lego, Language, and the Life of Bacteria. And it's going to be given by Professor Ansel Hsiao. He is an assistant professor in microbiology and plant pathology here at UCR. He received his PhD in cell and molecular biology from the University of Pennsylvania in 2008, and then he went on and did a postdoctoral fellowship at Washington University in St. Louis. He came to UCR in 2015, and his research here focuses on the identification of mechanisms by which bacteria in the gut or gut microbiota can actually be used to fight or establish resistance to other pathogens. So he's a real gut microbiologist, and he's going to be telling you a lot about bacteria and CRISPR and how that can be used in the future. So can you all join me in welcoming Dr. Ansel Hsiao to the podium?
All right, thank you very much for that introduction, Francie, and thank you for giving me the chance to give this lecture series. So, as you all know, this is the third in the lecture series talking about the use of these new technologies, specifically CRISPR-Cas systems in precision editing of DNA. And so what these CRISPR-Cas systems have done is really revolutionized our ability to rapidly and reproducibly modify DNA, which is the carrier of information in biological systems. Right? So by allowing us to modify DNA in new animal and plant systems, it allows us to change the fundamental structure and activity of organisms. Right? And so I guess being the third lecture, my job tonight is really to place the evolution of CRISPRs and the function of CRISPRs in the context of the system where it first evolved. Right? So CRISPRs are new from a research and industrial standpoint, but they evolved millions and millions of years ago in unicellular bacteria. And so that's what I'm going to focus on today. And I'm going to not really go too deep into the chemistry of it, being a lapsed biochemistry major myself, so we'll try to keep that fairly straightforward, okay? And so, from the very broad to the very specific, okay? And so, there we go. Okay, so organisms in general, okay? Cells, multicellular organisms, fundamentally are based on molecular building blocks, okay? So I'm going to take this Lego metaphor very literally for the entire talk. Okay, so these molecular building blocks are things like carbohydrates, proteins, nucleic acids, and lipids, okay? And organisms spend a huge amount of energy assembling these basic building blocks into more complex structures. So energy taking a mass of building blocks into something much, much more complex, okay? And so most of the energy of the organism is spent actually maintaining this structure, okay? And if you, like me, live in a household with a lot of small organisms that play with these things, you'll understand how easy it is to transition into the system on the left compared to the system on the right. Okay. So the cell takes these building blocks, assembles them in a very specific way in order to give itself structure and function. Okay. And part of how it does this is that it needs to be able to transfer information. So the cell needs to be able to understand, as it grows and as it multiplies, how to make additional copies of itself and how to assemble all of these building blocks in a very specific way. Okay? And so we can think about this flow as the movement of information, how to assemble cells into the final structure and activity. Okay? The particular configuration of all these building blocks and how that allows the cell to function. Okay? And this information flow transfers from cell to cell within the same generation, but also has to be passed on from a parent cell to its offspring cells, or from a parent organism to other organisms. Okay? And so you remember from your very basic science that the ability to carry information in biological systems is down to nucleic acid molecules. So DNA organized into genes. Okay? And so we can think about DNA as a blueprint. Right? So the particular configuration of individual DNA molecules within genes allows the cell, or allows an organism, in order to build all of these tools that allow the organism to perpetuate itself. And so as the DNA is replicated, this information flows from one generation of cells to the next generation of cells. Within a cell, the DNA can be turned into this messenger molecule called RNA via a process called transcription, which pretty much tells the overall cellular machinery, the machinery of the cell that allows it to maintain itself, to make proteins. Okay? And specific proteins are encoded by specific genes in the form of DNA sequences. So we can think of proteins as the tools that the cell makes in order to manufacture more of itself and to maintain itself under different environmental conditions. Okay? So structure and activity. Now, this is why we care about how we can modify DNA, right? Because if the DNA of the cell is the blueprint by which the cell makes the tools that it needs to assemble itself and to assemble the next generation of cells, then by changing the structure of DNA, we can change the structure and therefore the function of the proteins that DNA tells the cell how to make. Okay? So by changing the blueprint, we can change the resulting tools of the cell, and we can use it for our own benefit. Okay? And so gene editing, we can group that under a broad class of mechanisms for the modification of DNA. Okay? And so these other terms that I'll use, you might have heard of already, are basically just broader and broader subsets of that. Okay? So we can edit genes in the simple form of changing one base pair or one amino acid within a protein, but we can also remove the ability of the cell to make a certain protein, okay? Or we can introduce new proteins to the cell. And so, but we can also take large chunks of DNA from different sources and we can combine them. Okay? This so-called recombinant DNA technology, which I'll talk about in some specific detail later. Okay? And then going broader, we can use recombinant DNA techniques, gene editing techniques, in this process that we call synthetic biology. So we can construct completely new biological systems, or biological components, biological assemblies comprised of proteins, nucleic acids, etc., for our own benefit as humans. So DNA, okay, you all know DNA is a double-stranded molecule, and it's comprised of a particular sequence of these nitrogen bases. Okay, so T, A, C, G. And so the particular order of these individual nitrogen bases determines the information that the DNA carries. Okay. And I promised that I would keep the biochemistry straightforward, so we're going to go to a much more schematic view of how DNA works. Okay, so we have two strands of DNA, and then attached to those strands are these individual nitrogen bases that fit together in a particular way. Okay, so you can see, I think we've all played with these before, right? So the system on the right, you can imagine that these two strands will fit together in a very particular way to generate a stable structure of DNA on the right. And that means that because the individual order of these bases, of these nitrogen bases within DNA, affects how the DNA molecule fits together, small changes can actually have dramatic effects. Okay? And so we can think about this in terms of the communication of information, right? So if DNA really is a body of information that the cell accesses in order to make all of the tools it needs to respond to different environmental conditions, changing that information can really change the output. So we can think about it in terms of words, right? So GAG to TAG, if these were words, we change one letter only, that dramatically changes the meaning of the output. Okay? And so, I mean, these happen to be the letters that we use to identify nitrogen bases in DNA, but we can think about this in a more general term. So, for example, if you take 411 and dial it into your phone, you would get a dramatically different result than if you dial in 911 to your phone. Okay? And so this has a structural basis, right? So if we change individual components of the DNA, we have structural changes in the DNA as well. Okay? So on the left, you can see a DNA molecule is double-stranded. All the bases fit together in a particular way. Okay? However, we're going to change one base on the right in this structure. And so again, we've all played with Legos, I think. You can imagine that when you try to assemble the structure on the right, it's much, much less stable than the one on the left. Okay? So the individual sequence of these nitrogen bases within DNA really determine how the molecule comes together, okay? But it also defines a location code for a particular part of DNA. So in this case, we only have a sequence of 4 bases, but now we can talk about genes in terms of hundreds, thousands of bases. And so a particular sequence of individual bases really defines a very specific portion of the collection of all of the genes within the organism or the genome. Okay? And so that means that we can use nucleic acid molecules that fit into a certain way to the genome in order to target specific parts of the genome. Okay? So now, if we just look at one section of DNA, we have, let's say, a probe molecule, okay, a single-stranded nucleic acid. You can imagine that it's going to fit very well on the site on the left, but not so well on the site on the right. Okay? And so this is just, again, in schematic form. Okay? So we have DNA molecule double-stranded. These strands come apart, or can come apart. And now, if we have a molecule that specifically targets a particular order of base pairs within the DNA, we can bring those two molecules together. We can bring that portion of the genome into direct contact with this probe that we have introduced into the cell. Okay? And so that means that as this particular nucleic acid probe comes into contact with the genome, anything that's complex with it, whether it's a protein or another molecule, will also be brought to that specific site of the genome. Okay. And so we can take advantage of this in CRISPR-Cas editing technologies. Okay? The Legos will come back, so we'll do this slightly more detailed form. Okay? So now we have a particular target. Okay? So we have a particular gene within the genome or a particular location within the genome. This comprises, it defines order of individual base pairs. Okay. And we have a small molecule called a guide RNA. So this is a single-stranded nucleic acid. So it has all of those binding sites open to complex with one of the particular regions on the genome, defined by a particular DNA sequence. Okay. And this guide RNA is complex to a Cas family protein. Okay, so the Cas protein is the part of the CRISPR-Cas protein that actually has the mechanical activity on the DNA. Okay, so this synthetic RNA is designed to base pair specifically with a defined portion of the genome. Okay, so in Lego terms, we have this target that we want to hit within the genome. Okay, we can design an RNA probe that is going to bind specifically into that region. Okay, and this RNA probe, when bound to this Cas protein, this Cas enzyme, will bring the enzyme to that DNA, and that Cas protein will cut specifically at that site. Okay? And so this is a way of introducing a nick or break into the DNA structure at a site that is defined previously. Now, cells don't like this. Okay, so cells do not like breakages in the basic DNA structure. And so pretty much all cells have mechanisms that they use in order to repair these breaks within DNA. Okay? So turning a double-strand break into an intact linear molecule of DNA. Okay? And so we can take advantage of this in order to introduce changes to the DNA. Okay? So depending on how this double-strand break that can be made by cutting DNA is repaired, we can now introduce different bases into the DNA structure. Okay? So for example, if we provide a particular template of DNA, a particular molecule of DNA that is present at the time of the repair of this damage that's caused by the CRISPR system, the cell, the normal recombination mechanism of the cell, can introduce these sequences at a very specific site within the genome. Okay? And that means that we can introduce DNA and we can modify on a site-specific basis individual bases within the genome. Okay? And so I think now I want to transition into where this system came from. Okay, so CRISPRs, as you know, if you've been present for the last 2 lectures, you'll know that CRISPRs have been enormously useful, or at least enormously promising, in terms of our ability to modify organisms at the genetic level. Okay, and so it can be used potentially for human cells, but definitely for animals, plants, fungi, and bacteria and microbes. Okay? But I think it's important to remember that CRISPRs were not designed in a lab. So certainly we have refined the CRISPR system. We've adapted it to be very easy to use within research and industrial settings, okay, in the lab. But fundamentally, CRISPRs evolved in unicellular microbes. Okay? CRISPRs were not developed to allow us to modify DNA in a specific way. Okay? But these CRISPR-Cas systems that evolved in bacteria are really just the latest in a large number of different molecular systems that we've co-opted from bacteria and co-opted from microbes to help us perform particular biological functions. In this particular case, the modification of DNA. Okay? And it's not surprising that we derive a lot of potentially useful systems from bacteria, because when we look at biodiversity in general, okay, the diversity of genes, the diversity of organisms, most biodiversity in the world is actually microbial, specifically bacterial. Okay, so I remember from the first talk, there was this tree that identified particular relationships between individual organisms. And I want to say that was a little misleading in terms of how much diversity is present in the world as a microbiologist. So maybe I'm a little biased here. Okay? So in this tree, we're now looking at gene level differences. So we can look at a gene that is required basically in all cellular life, and we can look at how different that gene is between individual organisms. Okay? So in this particular diagram, if 2 particular endpoints on this diagram are close to one another, that means that they're related. They're highly related. Okay? Now you'll see that. So you'll see that eukaryotes are down here. Okay? So eukaryotes include all animals, all plants, and all fungi. Okay, so everything from unicellular brewer's yeast to blue whales are down here in terms of biological diversity. Okay? So you can see that the rest of this tree is really dominated by microbes. Okay? So especially bacteria, which were the first microbes to evolve, but also sort of these related organisms, archaea. Okay? And so bacteria have been around for a long time. They're the first organisms to evolve on Earth, we think. and they're found in pretty much all environments where we see life on Earth. Okay, so it's not too surprising that these microbes have evolved many different ways of dealing with interesting biological questions. And it's not too surprising that we, as we've studied the microbial world, have taken some of these systems for our own purposes. Okay. So bacteria and humans have a long and storied history. Okay, so sometimes this interaction is very bad for us. Okay, so for example, this disease that's known as the Black Death, okay, was a bacterial disease that devastated human populations in Europe in the 14th century. So maybe caused the death of about a third of the people in Europe at that time. Okay, and that was a bacterial infection. So that was one particular bacterium that caused the disease in humans, and that obviously was an interaction that was particularly harmful to humans. Okay? But that doesn't mean that bacteria aren't useful as well. Bacteria provide us with other important things. Okay? So a particular form of bacteria is critically important for malolactic fermentation, secondary fermentation, and the production of red wines. Pretty much all red wines, and I think a good proportion of white wines as well. Okay? And so bacteria can provide us with food, but bacteria can also harm us in their interactions. But also, I think just as importantly, is this notion that we exist, humans, animals, really exist as a complex of our own cells and bacterial cells. Okay? So humans are colonized by trillions and trillions of bacterial cells, okay? Especially in the gut, but pretty much on many different body surfaces, on your skin, within our oral cavity, within our respiratory tract. Okay? And if you count all of these cells, okay, these cells are obviously much smaller than our own cells, but it's thought that collectively all the microbial cells in the body dramatically outnumber your actual human cells. Okay? And so when we think about that, we're now talking about, for a normal human, on the order of pounds of bacterial cells, okay, which is not a unit that's typically applied to quantities of microbial cells, which suggests how dense these communities can be. And so it's not too surprising at that point, given that we know that microbial diversity is extremely broad, and there are so many microbes that are present within humans, and all of the genes, the unique genes that are contained with these microbes, dramatically outnumber the genes within our own genome. All right? And so one way of looking at this diversity is in the sense that microbes exist as complex communities. So it's not like in the lab where you have one type of bacterial cell that's growing in this nice nutrient-rich environment in pure culture. Okay. Microbes in the environment, whether it's in the human or in a bog, for instance, they exist in very complex communities. And so this is a visual look at how complex bacterial communities can be. This is a look at gut communities. Okay. So bacterial communities within the gut. And so all of these fluorescent points are individual bacterial cells. So you can see how densely colonized the portion of the gut is here. Okay? And each individual bacterial species is marked by a different color. So you can see that really these bacteria are distributing throughout the space, and they're now interacting with many different kinds of bacterial cells, many different kinds of other bacterial cells, as well as the cells of the host around it. Okay. Now, we can look at this diversity in genetic sense. Okay, so we can look at the genes that are present within bacterial genomes. So here we can look at how the genes within 3 different types of E. coli, Escherichia coli, overlap. Okay, so Escherichia coli, E. coli is this very common bacterium that you've probably actually encountered at some point in your science education, but there are many different kinds. Okay, so these E. coli's, depending on what kind they are, mediate very different functions. So over here, this dark circle is K12. This is the type that you might work with in your high school biology lab and so on and so forth. It doesn't cause any type of problems in humans. But then now we have bacteria that interact very differently with humans. Okay, so over here, this is a type of bacteria, the type of E. coli that causes urinary tract infections. And down here is O157H7 E. coli. So these are, you might have heard of this type of E. coli as a bacterium that causes food-related illnesses in humans, food poisoning, bloody diarrhea, and so forth. And so very different interactions. And so we can see that when we look at what types of genes are present. Okay, so these 3 types of E. coli, they're the same species of bacterium. they really only share about 40% of their gene content. Okay. Now, to give a point of reference, two closely related animal species, chimpanzees and humans, share over 96% of their genetic material, okay, in terms of DNA. So 4%, 2 to 4% difference between chimpanzees and humans, upwards of 60% differences across even one species of bacterium. So you can imagine how much overlap there is between very distantly related species of bacteria. Okay. And so huge amount of diversity in terms of the gene content of organisms within the bacterial group, and also in terms of how many species there are within the bacterial group. Okay. And so where does this diversity come from? Okay, so part of this diversity simply comes from the fact that bacteria have been around a very long time on Earth. And so they've had a chance to accumulate many different mutations as their DNA has replicated. Okay. Another way comes down to how bacteria interact with their environment, how they can acquire information from their environment in a sense. Okay. So to really look at that, I think we should talk about how bacteria can transfer their genetic information from generation to generation. Okay? So this is a video of bacteria dividing. Okay? You've probably seen something fairly similar. So an individual bacterial cell will replicate its genome and then will roughly double the size of the cell. And then the bacterial cell will septate and then split into two daughter cells. So each daughter cell now has an identical copy of its genome. Okay? Now, obviously in detail, this is quite different from reproduction in humans, okay? But in principle, we're talking about the transmission of genetic information in terms of DNA vertically. So we're moving from an older generation of cell or organism into the offspring, okay? So in this case, the variation in DNA is probably going to be down to things that mutate the DNA, but at a very low rate, maybe a few base pairs across the entire genome. But bacteria live in a complex environment. Okay, so they live in a complex environment in terms of the types of cells that are present, the species of bacteria that are present, environmental conditions. But because they live in complex environments with many bacterial cells, they effectively also live in a DNA-rich environment. So as bacterial cells grow within their environments, they're subjected to some stress, and not all bacterial cells will survive, right? So bacterial cells are constantly turning over in their environment. They're releasing their DNA into the environment. Okay? So the bacteria that do survive are surrounded by DNA from different species. And that DNA can be taken up by bacterial cells by a variety of mechanisms. So bacterial cells can grab DNA from the environment and bring it into the cell directly. Bacteria can, in some ways, also have sex. So here is an example of this process that's called conjugation, or bacterial sex. Okay? So you have a bacterium here that has made this long tubular structure. Okay? And this tubular structure, or conjugation pilus as we call it, connects the cytoplasm, the inside of this bacterial cell, to the inside of this bacterial cell. And through this conjugation pilus, DNA from this donor can be moved into this recipient cell. Okay? So bacteria, as they interact with one another, can actually transfer DNA from one cell to another. Now, some of this DNA that it either takes up from the environment or gets conjugated into the bacterial cell, probably just going to get degraded for food. DNAs are nucleic acids, they can get degraded for nutrients by the cell. But some of this genetic material will contain active genes. They'll contain functional genes. And those genes and the proteins that are encoded by those genes can confer a lot of benefits on a given bacterium. Okay, so in cartoon form, this is vertical transmission. So the genes and the genome of this cell gets replicated and it gets duplicated down to subsequent generation. When we talk about genes that are acquired by bacterial cells from their environment or from other bacteria, this is really horizontal gene transmission. So the movement of genes across, within the same generation of cells, within the same generation of organisms. Okay, so this bacterium is releasing this particular set of genes into the environment. This other bacterial cell is now taking up this DNA, whether by, in this case, it's not a direct interaction, but it could be a direct conjugation interaction, direct transfer into the cell. Now this cell will incorporate some of these genes into its genome and and this cell now is fundamentally different. It's changed its gene content. Okay? Now, when we think about reproduction in humans, this is absolutely not what happens, right? So we provide our DNA, recombine it somewhat, and give it to our offspring. The humans that we have offspring with, we are not exchanging genetic material and modifying our genomes at that level. Okay? So the genes that are acquired by these bacteria via horizontal acquisition of genes or horizontal transfer of genes can be retained if it gives some type of advantage. Okay? So we can think about it in perhaps this sense. So if I'm a bacterial cell and I am interested in building something in a given environment, okay? So I need to drive a bunch of nails into wood. But all I have is a screwdriver. It's not particularly useful for this task. Okay? But my neighboring cell has a hammer. And so I can acquire, maybe not so much the hammer if we're pushing this metaphor, but at least the knowledge to build the hammer from my neighbor. Okay? I can pass on this information to my children if they ever need to drive in nails someday. Okay? So these genes that may confer some type of temporary advantage to the cell can then be passed on to the offspring by subsequent vertical transmission. Okay? So bacteria acquire DNA from the environment. And sometimes this is particularly useful to bacteria, sometimes it's not. Okay? So genetic information can be transferred from the environment, DNA can be taken up or transferred from bacterial cell to bacterial cell. If they contain active genes, and those genes have some benefit for the bacterium, they'll retain those genes and then pass them on. Okay? But sometimes this communication in terms of genetic information transfer is particularly bad for the bacterium. Okay? And this happens when a bacterium is affected by a pathogen, another thing that causes a disease within bacterial cells. Okay? So I think it's important to remember scale in biological systems. Okay? So bacteria are very small compared to humans, but there are other things that are much smaller compared to bacteria. Okay? And so bacteria can be parasitized or these viruses, okay, can cause disease in bacteria. And viruses that cause disease specifically in bacteria are known as bacterial phages, or phages for short. So, they have these, particularly interesting geometrical shapes. But literally, the name basically means something that eats bacteria. Okay? And this interaction begins with a transfer of genetic information. So these phages, these phage particles, okay, attach to the surface of bacterial cells, and you can see these green lines. Okay, this represents the DNA that's contained within these phages being directly injected into the center of the bacterial cell. Okay? And this has some dramatic consequences for bacteria. So we're pushing technology by introducing YouTube into a PowerPoint. Okay? And so, yeah, I know. So here's a cartoon that illustrates what happens. So the phage injects its DNA into the bacterial cell. This DNA contains numerous genes. Okay? And the gene products basically hijack the machinery of the cell in order to replicate the phage many, many times. So the phage genome is replicated by these proteins. These proteins also make more copies of the structural protein of the phage. Now, eventually, as these phages multiply, they'll strain the structure of the host cell, and the host cell will burst, releasing these newly produced phages to affect other bacterial cells within the environment. Okay? So this is a particularly disastrous form of genetic transfer from the environment into a bacterial cell. Okay? But not all phage infections are going to be lethal to bacteria. Okay? So here's an example where a particular kind of phage will inject a gene that causes a bacterium to glow green. Okay? And so in this example, there are going to be two events. Okay? So we can look at down here and up here. So over here, you can see that the phage has injected a gene, okay, that caused it to glow green. However, in this particular example, that cell burst. Okay, so this is a time lapse, not real time. But in that particular example, the phage injected its genome and it caused the lysis or the destruction of the bacterial cell. But down here, Let's show that again. Okay, so here, phage is infected, and now the bacterium has been destroyed by the replication of that phage. But down here, the genes have been transferred into the bacterium. Okay, these bacteria having now retained some of these genes, okay, and remain glowing green. So they survive. They survive that particular infection. Okay? And so, given the dramatic effects of foreign DNA, specifically phage DNA, on bacteria, it makes sense that bacteria have evolved a number of mechanisms to control foreign DNA. Okay? So foreign DNA can be helpful, but they can also be very harmful. And some of these mechanisms are things like CRISPR-Cas. Okay? So now we go back to CRISPR-Cas. And so continuing with this Lego motif, I now have this nice Lego phage, okay? And it's coming to infect a bacterial cell. And so it's come to this bacterial cell, it's attached to the surface, and it's now injected its genome into the bacterium. Okay? But let's assume that in this particular case, the bacterial cell has survived the infection. Okay? So what can the bacterial cell do? Well, the bacterial cell can take this phage DNA, okay, and incorporate it into its own genome. So in this case, it has now taken a little bit of the phage genome and now incorporated it into a particular locus within its own genome. Okay? And so this actually is now the CRISPR is in this CRISPR locus. So hence the other part of the name. We have CRISPR, CRISPR-Cas. And so what does this allow the bacterium to do? Well, this allows the bacterial cell to remember that it has had an interaction with this phage as a foreign DNA. How does it do that? Well, the phage DNA is a specific sequence of individual bases, and those individual bases are now present within the genome of the bacteria that survived that first infection. And so if this bacterium, which had been infected but survived and incorporated a bit of the phage genome into its own genome over here, encounters this phage DNA again, what it can do is it can transcribe guide RNA. Okay? And so this guide RNA is complementary, or it fits into the structure of this phage genome. Okay? And the bacterium makes a Cas protein, a Cas enzyme that complexes with this guide RNA, and that's what allows this protein to be brought to the target, which is the phage genome. And this Cas protein, just like in the previous cartoon, will cut that phage DNA and now render that phage inert. Okay, so once the phage DNA has been disrupted in this way, it's no longer able to make complete proteins, take over the cell, and cause the death of the cell by rampant production of new phage. So an indication of just how important these mechanisms for controlling foreign DNA is for bacteria is that we found a number of them. So CRISPR-Cas is one of the more recent ones that have been characterized. Okay? But there are other protein systems, other biological machines within bacterial cells that control foreign DNA. And one of them or a group of them are these so-called restriction enzymes. Okay? So just like Cas, they are nucleases. They will cut DNA. But they cut at very specific sites. Okay? And so oftentimes they'll recognize a very specific DNA sequence, and they'll cut in a very specific pattern. Okay? So for example, this enzyme generated this overhang within the DNA. Now these enzymes were first discovered in the context of control of phage. Okay? So in the 50s, okay, it was noticed that there were some types of E. coli that were particularly resistant to phage. Okay? So they restricted the growth of phage. And it turns out that this was based on the fact that those types of E. coli expressed these so-called restriction enzymes. So these enzymes were able to cut and cause the degradation of phage genomes that were injected into those E. colis. Okay. Now, you might ask, well, these restriction enzymes, they cut at a particular site of DNA. This probably occurs at multiple points within the host bacterial genome. So how does the host prevent itself from digesting its own genome? Okay, and so one way of dealing with it is that the host DNA is chemically modified, okay, by the addition of these, little chemical groups that get attached on to the backbone of the DNA. So the host DNA will have these protective groups, and so the enzyme will not cut this. But foreign DNA that comes in will not have these protective biochemical modifications, or they'll have a different pattern of biochemical modifications, and that foreign DNA will be digested. Okay. So restriction enzymes discovered first in the 50s, the action of restriction enzymes first discovered in the 50s, and then the first individual enzymes, the first proteins were characterized in the 60s. Okay. So what did that allow us to do? Okay, so we were able to isolate these enzymes as time went on and research continued. Okay, so we were able to use these enzymes to generate basically DNAs with compatible fitting ends. Okay, that allowed us, for the first time, to put different DNAs together in a very specific way. So you can think of having two DNA fragments that we might want to join together. Okay, and they both have a specific set of bases in DNA form at the end of them. And so if you have a restriction enzyme, it will cut at these two sites, generate an overhang. Okay, so now these are compatible ends. And you can imagine what will happen when you put them together. Now we were able, in the 70s, okay, to combine individual DNA fragments together in a controlled way. And this was revolutionary. So this allowed us to begin doing recombinant DNA techniques. We can take DNA from different sources, put them together in a manner of our own choosing. Okay? And so at this point, in the 70s, we are doing gene editing, only not with CRISPR-Cas systems, okay, but with restriction enzymes, which ultimately come from the same need of bacterial cells to control foreign DNA that are introduced. Okay. So an example of how recombinant DNA was used that may have affected some of your lives specifically is the production of insulin. So does anyone take insulin in this room? No one. Okay, maybe a few people. All right, so insulin is a human protein that's really important for our ability to control our blood sugar levels. Okay? So individuals with diabetes, well, type 1 diabetes, don't make sufficient quantities of insulin. And so they have a hard time controlling their blood sugar levels. So they need to take insulin in order to help them deal with that. Now, originally, insulin was produced, insulin ultimately is produced by cells of the pancreas. Okay? So before any of these recombinant DNA techniques, insulin was purified from animal sources. Okay? So an example is, this is from Genentech, this information. Okay? So before recombinant DNA technologies, to make 1 pound of insulin, purified insulin, required about 8,000 pounds worth of frozen pancreas. Okay? And that would have been derived from thousands and thousands of animals, pigs and cows. Okay. So what we've been able to do, and we as in kind of the royal we of science, is that we've been able to take the insulin gene, okay, the blueprint that allows human cells to make this insulin protein, and put them into a bacterium. And now we can have bacteria or microbes make that protein for us. So how do we do that? Well, we can take the human insulin gene in the form of DNA. We can cut it via these restriction enzymes. Okay? So we can identify sites where these restriction enzymes can cut in a specific way. And we can introduce, with those compatible ends now, we can take that gene and insert it into a bit of bacterial DNA. So this bit of bacterial DNA, called a plasmid, is able to replicate within a bacterial cell. So now we've generated a recombinant DNA. We've taken DNA from two sources. Okay, a self-replicating piece of bacterial DNA and a gene that encodes for a human protein. We can now put this construct into a bacterium and grow it very rapidly under industrial processes. And then the resulting recombinant bacterial cells, which now have this edited version, okay, of the human gene, is now able to produce human insulin protein. And so we can take these large batches of bacteria, and now we can purify insulin. Okay? And so what this enabled is basically a 50-fold production increase in insulin. Okay? And so the insulin that was produced in a much more cost-effective and easy way. It had similar activities when introduced into humans. And this insulin was actually of increased purity. So we no longer had to purify them from bulk tissue from animals. And that also avoided some of the allergic problems that were generated from taking animal proteins in humans. And so this work dated from the 70s. Okay? And so some of this work was actually done at City of Hope locally in collaboration with Genentech, which at the time had 12 people, and now it's much bigger than that. Okay. So, how fast are we going with these with these DNA modification techniques in bacteria specifically? Okay, so we talked about an example in which a system that was designed, that evolved in bacteria to control foreign DNAs, just like CRISPRs, was used to generate genes for our own purposes, for our own industrial purposes, industrial uses. Okay, but that was one gene. There's one gene into one plasmid. What can we do now? Well, now, well, maybe not now. In 2010, okay, J. Craig Venter Institute published a study in which they claimed to have generated, and they did generate, a synthetic bacterial cell. Okay, so what do we mean by a synthetic bacterial cell? Now, instead of putting together little bits of DNA, one gene at a time, they were able to chemically synthesize many different genes from a very small bacterium, okay, put them together, again, purely chemically, okay, not involving an individual bacterial cell. So they made an artificial genome with these recombinant DNA techniques. And that artificial genome was then inserted into a bacterial cell. So when that bacterial cell divided, it made a copy of this synthetic genome. Okay? And it vertically transmitted this synthetic genome onto the next generation. And in that next generation, they were able to isolate these individual synthetic cells. Now they're synthetic because the blueprint that controls how they function, how they divide in subsequent generations, were derived from rational design. Okay? So we, for the first time, were able to totally control the total genome of a bacterial organism. Okay? And so that's an indication of how fast these DNA techniques are evolving, okay? And how many useful roles we've been able to find for these individual enzymes in bacteria. Okay. To sum up, gene modification in bacteria. So technologies, CRISPR-Cas technologies, restriction enzyme technologies were one of many systems that were evolved by unicellular bacteria for DNA modification themselves. Okay? And that's because they live in complex environments, they're constantly taking up DNA, and they need ways of controlling the function of that foreign DNA. Now, I think that CRISPR is not going to be the end of it. Okay, so you remember the enormous genetic diversity of microbes. I think that as we continue to study the microbial world, we're going to be able to identify additional mechanisms that we can put to other uses. Okay, so just like we co-opted CRISPR, a phage defense in bacterium, for precision gene editing in animals and plants and other bacteria, I think that we're going to find other systems that allow us to do even more interesting things to biological systems. Okay? And so CRISPRs are not also going to be the end in terms of our ability to study microbes. Okay? So when I talk about putting together synthetic microbes in these small bacteria, not all bacteria are understood well enough for us to be able to modify genes at that level. And so because CRISPRs are so straightforward, we have a guide RNA, we have the Cas protein, and then we have an intervening piece of DNA that is incorporated at that double-strand break. This might actually allow us to go in and target genes in bacteria that we don't understand very well, that we don't have a lot of other tools to manipulate their genomes. And so it's possible that this discovery that protected bacteria from the activities of phage can help us open up the world of bacteria for further study. Okay? And that's pretty much what I have. So I'd be happy to take any questions.
Thank you, Ansel. That was actually a fabulous talk. We're going to sit over there. We'll take questions. We'll let you guys get lined up. I'll repeat the questions so that we can hear it in the recording. And while we're getting organized, I'd just like to point out that old paper that Ansel showed you of insulin being expressed in bacteria, that was done by a UCR graduate. Art Riggs, who is the lead author on that study, is now at City of Hope, was a undergraduate student here at UCR. So sort of comes full circle.
You're going to use that?
on. Okay, it's on. Okay. Okay, I think we'll maybe start at this side here while the other side's getting set up.
Hi, so my question is, can gene modification using CRISPR result in a new domain in the phylogenetic tree of life?
Okay, great question. So the question is, could gene editing using CRISPR result in a new phylogenetic domain in the tree of life?
Yeah, that is a very good question. I think that as somebody that works with different populations of bacteria, identifying different populations of bacteria, this discussion has come up in that, so we define bacteria in terms of how similar key genes are, right? But that gene is not necessarily determinative of how that bacterium is going to function, right? It's just a marker. And so just like we say that, well, different types of E. coli only share about 40% of their genome, but they're all the same E. coli, while we're using a very limited set of genes to determine that it's an E. coli. And so to a certain extent, yes, we can introduce new taxonomic divisions of bacteria simply by editing that one gene that everyone uses as a marker. But I think that's just an indication that, well, it's really the whole genome. It's not one particular gene that defines a species and therefore defines a function. It's all of the other genes that are present in the right combination that determines how the bacterium is going to function.
Thank you.
Yeah.
And over on the side over here.
Hello. So I was wondering, when we do CRISPR, how do we make sure to get the, how do we get into the nucleus when it has to get through the ER and the Golgi apparatus and vesicle transports?
Okay, so the question is that when we use CRISPR in eukaryotic cells, how do we get the CRISPR-Cas9 and the guide RNA into the nucleus of the cell, and how does it avoid the endoplasmic reticulum and the Golgi, for example?
Well, so the ability to control where these mechanisms go is actually a really open question in terms of CRISPR, right? So in principle, if we can get just the guide RNA and the protein and the nuclease protein into the cell, we should be able to have a fairly reasonable amount of success at targeting a particular site. But in terms of getting those materials into the body, right? So let's say that we're interested in modifying a large number of cells within a multicellular organism. That's actually a question that I don't think has really been fully addressed, right? So this idea of penetrance, what proportion of cells can we modify at any given time is actually a very important one. And I don't think we have a good answer for that yet. So much in the same way that if you take a drug, we don't know if that's necessarily going to go into the right place or into all the places that it needs to go.
I'll just add that the DNA that we use to make CRISPR and Cas9 is no different than any other DNA. You can take a plasmid, one of those circular circles of DNA, and you can put it into any organism, and it will get into the nucleus. It's just sort of what it does, right?
Thank you.
Okay. Do we have a question over here?
Oh, yeah.
Danielle?
I was wondering, I saw that totally synthetic bacteria that we were talking about, and I'm confused as to how it's totally synthetic, because you start with, I want to know exactly what they did. Did they knock out genes, the original genes, and they got the plasma membrane from somewhere?
Right. So
the question is, I'll take time to think about it. So the question is, how did they get that synthetic bacterium? Was it really totally synthetic? For example, where did they get the plasma membrane to put the DNA into, et cetera?
Yeah. So let's be open about this. There is a reasonable amount of marketing involved, right, in how JCVI has chosen to present this. You actually make a very good point. It is not fully synthetic in the sense that the machinery was provided synthetically, right? So they didn't synthesize all the ribosomes necessary to make proteins and et cetera. So they basically needed a live cell in order to jumpstart the mechanism. Now, but because the genes that were provided, right, formed the template for making subsequent generations of these cells, after that point, right, as this synthetic cell is using this synthetic genome to make all the different parts of itself. Eventually, those original components are going to get diluted out by the division of the cell. And then you'll actually reach the point at which most of it is truly synthetic. Now, this is an interesting evolutionary question, right? We don't really understand how the first membrane formed around the first nucleic acid that transferred information. Right? A great question. Yeah.
Okay, question over here.
Sure. So thank you very much for the talk today. I thought it was very interesting. So to get back to the synthetic biology question, and the chimpanzee humans are 96% identical in terms of their DNA. Could you take a human genome and edit all the parts out that are non-chimpanzee-like and get yourself a chimpanzee out of an original human genome, or is the amount of CRISPRing that you'd have to do that just too much? Like, are you getting into the, like, what are our technical limitations with regards to multiple edits that would require huge amounts of cutting and pasting across the genome?
So the question, if I think I got it right, would it be possible to use CRISPR to edit out all the uniquely human genes and then revert back to a chimpanzee? Is that the right question?
Wouldn't necessarily be a chimp, it would be a chimpanzee, because it would be
Ch-
just starting with
chimpanzee,
human
okay, love
DNA
it.
and just trying to get a chimpanzee out of it. So, but
great question.
It's really a technical, like, how technically advanced are we right now, or is that just beyond the realm of possibility, or is that 10 years from now, 3 years from now, 2 years ago? We already did it. I don't know about it. But.
Okay. Are there any chimpanzees in the audience? I think that's what he's asking.
So we definitely haven't done it, and I don't think we are anywhere close to it. And so, I want to talk a little bit about the synthetic bacterial cell as an example first, and then we'll go to this particular human example. So this idea that we can generate a synthetic genome is not conceptually all that difficult, right? They simply picked a genome that was small enough, a starting genome that was small enough so that they could do it in a logistically reasonable fashion. And in the case of the bacterium, a lot of it is just willpower. Can we dedicate enough money, enough time in order to make all of these individual pieces and splice them together? Now, theoretically, we can go into a genome and CRISPR everything. It might take centuries, who knows, in terms of just the technical aspect of it. But I think in terms of what you're talking about, this idea that we can now just change all of the different base pairs and turn one species into another, well, we know that there's a certain amount of identity in terms of, like, okay, how this particular kind of DNA versus this particular kind of DNA, and how many base pairs match, right? But we really don't understand what all of these pieces of DNA do, right? So we can go in and maybe make a direct copy for certain parts of it. Logistically, it would be a nightmare for a human genome, right? But even in bacteria, so E. coli, which we've studied for since genes were discovered, E. coli we've studied, we're looking at, we have a guess as to maybe about what 70% of those genes do. And that's for an organism that's been studied intensively by thousands of people over decades and decades and decades. And so we're really not at the point at which we can, for many traits at least within bacteria, but definitely within more complex organisms, identify, well, it's this sequence that causes it, right?
Yeah, I'll just also add, even though the human and the chip genome is about 96% identical, so there's 4% difference, there's no gene in humans that is not in a chimp, and vice versa. There's no human-specific gene. The differences that are there often, it's just in between genes, it's in regulatory sequences, et cetera, right? So just the premise of that is not, and in fact, even between humans and mice. There are some genes in mice that we don't have in humans, but I would say well over 90% of the genes in human are also found in mice, and they work perfectly fine. So we're a lot more similar than one would expect. Okay. There's no other person there. Okay, all right, we'll go back to this side.
Hello again. So another question I was wondering if anyone had attempted CRISPR on mitochondria, since we believe their DNA is different from the host cell, from the symbiosis theory.
Okay, so great question. Has anyone attempted CRISPR-Cas9 on mitochondrial DNA? Because mitochondria originally came from a bacterium that was engulfed in a larger cell.
I don't believe that's ever been tried, Actually, it's quite, I think the field of CRISPR is exploding. It's possible that somebody has tried to manipulate the mitochondrial genome, but I'm not aware of a particular example for it. Yeah.
That's a good question. Okay.
So it's possibility of publication then.
Yeah.
All right.
Well, oh, yeah, first you got to do it. Yeah.
Thank you for the idea. Thanks.
Yeah. Thank you.
My question is CRISPR is it an effective device to be used to help create antiviral, basically devices for humans, I guess? And is that kind of a danger if we start giving bacteria just abilities to kill viruses it wasn't meant to kill? Like, is that something they're trying to do, or?
So if I got the question right, can CRISPR-Cas9 be used as an antiviral agent?
For humans.
For humans to somehow attack human viruses?
Yeah, so I think it's important to remember that when CRISPRs are used as an antiviral device in bacteria, it's really not something that it is acting away from the bacterial cell. Okay, so the virus needs to be able to come into, basically needs to be able to send its genome into the bacterium, and the bacterium needs to have seen that, needs to have had a bit of that genome in order to make the guide RNA to target it. So that means that viruses of humans are not really ever going to be able to infect bacterial cells, because viruses are actually very specific in terms of the cell types that they infect. So I don't think we're going to get to the situation of having a bacterium circulating within the human body that essentially sops up human viruses. Now, that said, I think that we can get to the point at which potentially we are modifying certain subsets of human cells to express certain antiviral proteins. That's within the realm of possibility.
Just as a follow-up question, so the CRISPR-Cas9 system, as I understand, is in every bacterium or every bacterial species that is looked at, but we definitely do not have it in humans. Could you explain why humans lost that ability to have a CRISPR-Cas9?
That's a good question. I actually don't know. So I think in terms of, I don't know enough about the evolution of CRISPR from bacteria. Yeah, it's a great question.
Well, I guess the thought, since we're multicellular organisms, we don't have phages or viruses affecting us, and so we developed a different, more complex immune system. Whatever. Okay, do we have another question over there?
Yes. This is kind of the other side of the coin of the chimpanzee-human question. We know about the current applications affecting diabetics and certain indicator genes for diseases like Tay-Sachs and sickle cell. What do you see in the next 5 to 10 years, relatively short time horizon, as practical applications of technology?
Okay, so great question. What do you see as the practical applications in 5 or 10 years for diseases such as sickle cell anemia or Tay-Sachs disease? Can we use
or any others?
Or any other?
Or really any other implications, not just disease fighting, but human enhancement or whatever you can think of.
Yeah, I think that probably the most widely accepted uses of CRISPR are really going to be modifications of basic industrial processes, right? So we can use them to modify cell types that have been used to produce various things of interest, and also definitely within the research field. So we can use CRISPR to modify things we weren't able to modify before in terms of bacterial species that we haven't been able to really manipulate. Now, in terms of humans, I think that I'm not a human biologist, but I think that in terms of humans, we're more limited in terms of our willingness to explore that possibility, right? So we can take subsets of, like even now we're taking subsets of human cells, we're isolating them from the blood, we're modifying them in some way and giving them back into a patient in order to treat a variety of things. So perhaps we can use CRISPR to speed that process up. But in terms of basically just targeting cells within our tissues, across all of our tissues, I don't think that's going to be done in the next 5 to 10 years. Yeah.
I have a sort of a related question. So we have all these microbes in our gut, and they're going to be susceptible. We could easily modify them by CRISPR. Do you see an application of using either CRISPR-Cas9 or some other recombinant DNA technology, or perhaps the bacteriophages themselves, to modify our gut microbiome to then potentially treat diseases, because there's a lot of diseases that are associated with imbalance in our microbial populations.
Yeah, I think that's definitely a possibility. So in terms of the bacteria that are present within the body, and this is not restricted to the gut, there are plenty of bacteria in the skin and in the oral cavity that we can't really modify, right? And so if we want to deliver therapeutics, for instance, in a site-specific way, we're going to need to be able to modify the genomes of these organisms. And before, we really didn't have tools for the vast majority of these organisms. So, in my talk, I talked about systems that have been well studied, things like E. coli, because that highlights what we're able to do with these technologies. But many bacteria, we don't have these technologies available, right? And so, yeah, I think in terms of introducing genes that provide some therapeutic benefit to humans, and having bacteria that have evolved really to live at the site where we want these therapeutics delivered is going to be a fertile area of research. So we can think about, I'm sure all of you have taken probiotics or live yogurts at some point. Well, those things may have health benefits, but they need to be continuously taken because these bacteria were not selected to live in the gut. They weren't. They were selected to do well in acidified milk. And so industrially this is great. It means that you have to keep buying probiotics in order to derive that benefit. But if we want to have a more permanent benefit, we would really want to engineer a bacterium that lives in the site, that has evolved to live in the site to provide that for us.
Another question over there?
I'm just wondering, what other tricks are there that the bacteria do that we can learn from besides the CRISP method that you've been just talking about?
Oh, that's great. What are the other tricks that bacteria can do that we could learn from in the future? Ansel mentioned first the restriction enzymes, and now there's this CRISPR-Cas9. What other secrets in the genomes are they hiding that we could potentially benefit from in the future?
Yeah, if I were able to predict the next breakthrough, I think that would be a great trait to have for me. But I can talk about some other things that we derive that are useful from bacteria, for instance. So a lot of from microbes in general. So a lot of, for example, the antibiotics that we take are actually microbially derived, right? They were also not developed in the lab with a chemist looking at the structure saying, well, I'm going to rationally design this molecule to target an organism. And this is not terribly surprising because of the complexity of natural bacterial or microbial communities. These organisms are living in close proximity to one another and they're actively competing against one another, right? So this is, I think, something that we can definitely start to look at in terms of bacterial genomes, identifying things that are an important part of natural bacterial evolution, the ability to compete with other microbes. I think recently, a few years ago, there was a completely new class of antibiotics that was identified from soil bacterium, for instance. Right? Yep.
Okay, all right. Well, if there are no more questions, I'd like for you to join me in thanking Dr. Ansel Hsiao one more time. And I hope you'll join us at the last lecture. It's going to be on May 10th. We're going to have a little bit of a gap. It's going to be Professor Carl Krainer, who is a professor of philosophy here at UCR, and he's going to circle back and to some of these basic questions in terms of how to use CRISPR-Cas9 in terms of ethics, et cetera. We will have the three speakers, and I will be here as well, the three speakers that you've already heard, and in case there's some other technical questions that come up. So we hope it'll be an interesting discussion. We hope you'll join us then. Okay, thank you very much. Have a good evening.